Perfect. So the next and the last presentation we have for the meeting is the Amphibians of Virginia, a book in progress, and that's going to be given by Dr. Jason Gibson from Averett University and Dr. Paul Sattler from Liberty University.
I'm not a doctor. I'm a mister.
No. I'd rather be wrong about that than do it the opposite way.
So Paul stressed me out. He says I got 6 minutes, so I'm going to go ahead and just kind of dive in. But Paul and I are, I guess, fools for working on a project on a book on the Amphibians of Virginia. Paul told me in 2020, as we started working on this, Jason, that's going to take about 10 years, and he was totally right. We were hoping Joe Mitchell was going to write the Amphibians of Virginia, but unfortunately he passed away. And then we were hoping Steve Robley was going to write the book on the Amphibians of Virginia, but he got interested in bugs. So it kind of fell to us to kind of do this. So we're kind of modeling our book after Joe Mitchell's book. And it took him 10 years to write the Reptiles of Virginia, as stated in his introduction. So our book is going to be basically species accounts of all the amphibians of Virginia, which is a lot. And so we're just basically going to have species accounts where we have descriptions and talk about male-female differences, confusing species, anomalies, just the biology, everything we know from the literature, everything that Paul and I have been doing, and from the VHS surveys. We're just trying to make a compilation of all that material and put it into one book. And it's been quite a hydra. So every time you slice a head off, two grow. So what we wanted to do today was to talk about the problems we're having, and then how can you help us? So one of our first problems, I'm going to talk about anurans, and then Paul's going to tag team, and he's going to do salamanders. So one of our first problems is, what do you even call them? So, and I'm serious. If you take the Frog Bible, which is Dodd's Frogs of the United States and Canada, he just basically took Hyla out and replaced it with Dryophytes, and took Pseudacris out and replaced it with Hyleola. I had to even phonetically pronounce it out. So he basically follows the American Museum of Natural History's naming sequence. But if you go to AmphibiaWeb, they basically use Rana and not Lithobates. Dodd uses Lithobates, and he uses Hyla. AmphibiaWeb, excuse me, uses Hyla and not Dryophytes. They do use Pseudacris and Anaxyrus. And then if you go to SSAR, who, kind of what the VHS website uses, they, in the Herpetological Review, use Buffa and Anaxyrus in the same issue. Okay, so two different genus names for the same things. They use Rana and Lithobates in the same issue. They use Hyla and Dryophytes in the same issue. So they don't really know what to do. And if you look at their website, they have and highlighted there, we are currently working on the 9th edition of the list, which is the new species list of names, and hope to have it published on our website by the end of 2023. And it's 2025 almost, and they haven't had that out. So we don't know what to call even anurans. And Paul will talk a little bit more about that when he talks. So our second problem is where do they live? Where are they located? So this is a really bad issue or good issue. I don't know how to look at it. But because of iNaturalist and because of all the species distribution ranges changing because of Catspiana, there's huge swings in range extensions that are going on right now. Making a range map makes me sweat because it's going to be obsolete as soon as it's published. So species, just from my own 25 years of looking at stuff, I've seen species ranges extending. Hylocinera, the green tree frogs, barking tree frogs. Barking tree frogs are showing up everywhere, and they were just taken off the state list of being a state species of concern, and now they're everywhere. I believe Steve even found the most northern range record just here recently, and we're hoping he publishes that in Catspiana soon. So, but he found one up in, I think, two sites in King and Queen County. So pine woods tree frogs, squirrel tree frogs, these are just expanding their range. I don't know if they're hitchhiking. I don't know if it's just because more people are looking, but it's definitely something we're seeing. The coastal plains leopard frog, southern leopard frog, whatever you want to call it. So that's showing up at fish hatcheries. So, so these things are basically expanding their ranges. Problematic distributions we're finding is misidentifications with the eastern cricket frog and southern cricket frog. So it's hard to rely on observations and published observations that have been made of these. At some places in Virginia, I can't tell the difference between American toads and Southern toads. They're almost indistinguishable, and they're probably hybridizing. So we definitely need somebody to do some genetics work on that to get those distributions worked out. Obviously, the gray tree frogs. The gray tree frogs are, they're gray tree frogs, and some people are misidentifying them based on call. And then my personal one that I've been working on is the eastern spadefoot, and I can't understand why in the world it's not in Virginia Beach. It's completely annoying me when it's found almost everywhere. And you can see there's a whole series of counties in the center in the Piedmont that we haven't found those at yet. Tadpoles, another nightmare scenario. We did get some feedback on one of our chapters, and one of the feedback was, where are the tadpole keys? Well, they're very, very difficult to do. So tadpoles, it's frozen. Let the gurus come. So tadpoles exhibit phenotypic plasticity. So if you go to a different habitat, they are going to look different. Sorry, you might just need to stand here and hit it. What are you doing? Clicking the button?
Just click that arrow.
Okay. It's tough. It is. It's tough. So if you take a different food type, a different predator, a different flow rate, a different substrate, they look different. So we have the eastern cricket frog on the left-hand side without predators, eastern cricket frog on the right side with predators. So basically, it's almost a fool's errand to write a key on it. These are 3 keys that are from from books. I won't even tell the names of them because I don't want to embarrass anybody. So the way that you identify tadpoles, according to these keys, are counting these little labial tooth rows. So in this particular one, they have 5, 6, or 6, 5. So 5 upper lips, 6 lower lips, or 6 upper and 5 lower. And they have a picture of 5, 5, which doesn't even fit their key. So then we have another identification key for North Carolina that has a formula of 2, 4, or more. I don't even know what or more means. And then in a relatively recent book, we have a species where it has, unfortunately, I made a typo right there. It should be 6, 6, but it has 6 uppers and 6 lowers, which doesn't fit any key. So that's just for the eastern spadefoot. So these are very, very difficult species to do. We have really good information on these particular very common species. We have very moderate amounts in these particular species. Here's where you can help. So that mid-Atlantic coastal leopard frog newly described, carpenter frog, Brimley's chorus frog, New Jersey's chorus frog, these we don't have a lot of data on. Thankfully, Evan Spears has told us about a good site for Brimley's chorus frog, and but we're still trying to get them, and they've been pretty problematic. That species right there, we know almost nothing about, maybe some distribution, but almost nothing about any of its natural history. Bullfrogs exceeded our scales that we were using. We finally got a bigger digital scale, so we're now trying to get some species, collected some data on those. Carpenter frogs. We have Todd over here. Todd's the world's leading expert in carpenter frogs in Virginia, but he used non-digital measurements, so we couldn't use his measurements. We're using digital measurements, so we're having to go out and collect stuff, and it's been challenging to get out there at the right time. But New Jersey's chorus frogs, Brimley's chorus frogs, that's data that you can help us with. We are trying to figure out the phenology, the life histories of frogs in all the different regions. So we broke Virginia up into different regions. So if you live in one of those regions that's a block up there, spend a year in your yard listening to stuff and then publish your results. Okay, that'd be very useful. We know that things are calling different, laying different numbers of eggs. They have different seasonal variations in those different regions. So we're trying to tackle, that for Virginia. All right, my friend, tag.
So on the salamanders, we've got a number of problems. How many species are in Virginia? The DNA studies are splitting various species at a very high rate. This is a list of species that are found in Virginia. All the ones in green have been added since Mitchell and Ray, the atlas, came out. The ones in red have been removed since Mitchell and Ray came out. Richard Heighton began a lot of these molecular studies with revision of Plethodon glutinosus, the slimy salamander. So he, based on electrophoresis, protein electrophoresis, split it into 16 species. More have been added since then, and morphological differences have been found after the isozymes showed where those different divisions would be. So those new species have held up over time. And the question is, will genetic discontinuities revealed by the DNA sequences hold up like the isozyme data did? Big question, are these genetic differences shown by mitochondrial nuclear DNA, are they at the species level, or is it just population differences? And will we find morphological differences to back up those morphological differences? Because we know virtually nothing about what's occurring at the contact zones. Oh, what's so this is Prian's work on Desmognathus fuscus, and you've got all these different types of fuscus that now are being split into different species. So this is a map here from salamanders of the eastern US. Now we've got the wolf dusky salamander along the southern tier of Virginia. You've got the Carolina swamp dusky salamander, where we used to say it was the southern dusky. Plethodon whirlii has been split into several different species. You've got Dixie at the Dixie Caverns and surrounding areas there, Jacksoni at the southwestern portion. Black belly salamander, this is from their molecular work. I don't know if you can see, but over on the side, you've got these different types of quadrimaculatus and marmoratus, the black belly and shovel nose salamanders, and they don't split out with all the black bellies together and all the shovel nose together. They're all mixed together. So this is from their new book, Quadrimaculatus. It's a cryptic species, as many as 6 genetically distinct but morphologically indistinguishable populations. And hopefully, well, they're assuming that there's reproductive isolation there. We will see. So now in Virginia, Quadrimaculatus type D is now described as Desmognathus canoa. Desmognathus acrophius and orestes are being split up further, so we may see additional new species coming there. And what about state databases? Where do we find the different salamanders? Are black belly salamanders really up in Amherst and Bedford counties? Don't think so. For the sake of time, I'm going to skip over this example. So we're having trouble just keeping up with the number of new species that have been described here in Virginia, and we don't really know how accurate the ranges are. Where can we go to find the different species? Do we need to use molecular techniques out for a particular population that we're working on, what it is? We need to obtain photos of the animals, we need phenotypic data, we need natural history data, which is almost lacking for many species. So what you could do to help us would be start taking field notes, complete field notes. When is it that a particular species first comes out in the spring? What is the temperature, air and water temperature? When do they start calling? When do they start breeding? When do you see the first egg masses? When do tadpoles and larvae hatch? What's the beginning of metamorphosis? So if you can keep records over a number of years, that would greatly aid what we know about Virginia species. Then, if we can put all that data together from the different regions within the state, we can hopefully start to understand when those events occur, and if they're changing over time, then maybe we'll be able to finish the book. So we can fish for questions if there's time.
And you're shifting all the responsibility of getting it done on everyone else.
We're asking for help.
Wow.
One question. Are you just looking for that information for your species of interest? Or are you looking for that information for any contested
Any amphibian in Virginia. Thank you.
All in case